brain cells from iPS cells as well. So we can also test different plasma systems on these to check the inflammation on these systems too. So actually for those as well, so in last 1 year I was not alone, so I had very brave, very motivated students in my group. So I really thank to them and all my funding agencies. They believed to our work. Hopefully we will be sharing more results on these projects soon too.
Thanks for the information you provided.
Thank you, Nurajam. I have one question.
Of course.
Well, mine is more technical, actually. So this organoid system, the cells share the same canal, right? Same channel. So they're fed by the same media, I assume. So how do you recover them if you want to do an ICC experiment or any other experiment for a specific gene, for a specific cell? How do you recover the cells?
Yes, you can basically, well, for any immunofluorescent imagings, I showed some of the confocal images that we took on the chips. The chips themselves are transparent, so we can fix the chips at the cells inside of the chip, so while they are interacting with each other, and then do immunofluorescent imaging. And also, if we need to do more like flow cytometer, Western blot, or like RNA-seq with these cells, we can actually collect the cells from the channels.
But everything together though, right?
Basically, the endothelial cells are separate and the brain cells together.
Okay, I see.
Because it has two channels. One channel is just for the endothelial cells.
They don't interact even with the small pores? They don't interact?
Well, they are interacting, but
they have to.
Like the collection purity of them quite efficient. And if we need to, we can actually sort them and separate them after collecting.
I might ask you more later because I'm also interested in microfluidic systems. So maybe in the future I can ask you more questions.
Of course, yes.
Thank you so much. That was good. I'm done with my questions. And as you can continue.
First question, is it possible to detect immunity levels with biosensors?
What? Detect what? Sorry.
Is it possible to detect immunity levels with biosensors?
Well, it is possible if you are incorporating any biosensor into the system, yes.
Thank you. Second question, if this organ chip application is applied everywhere and for each tissue, do you think animal use as objects will be completely terminated?
I did not understand the question, I guess.
I repeat that. If this organ chip application is applied everywhere and for each tissue, do you think animal use as objects will be completely terminated?
No, I don't think the animal models will be ended. Hopefully, that's the goal. Using these like functional units of the human cells, we would like to minimize the using of the animal models and sacrificing the animals for these type of research. But I don't think we will be eliminating them very soon because these models are recently emerging and they still need to be optimized quite well. And as you can see, like for the blood-brain barrier, I'm still like, if I would like to test a drug, I need to be able to test them in an entire organism too. So I'm missing all the other organ interactions. So that will take time. But in 10 years, we may be describing like a totally different things. And I will say, okay, this is possible. But right now, it will take time.
Thank you very much for participating. It was a very useful talk.
Thank you. Thank you so much for inviting me and it's funny. my time.
Thank,
In your time, actually, like this beautiful Saturday. Thank you.
thank you. Now a 10-minute breaks and then Görke Gürel Pekezer will be with us. Yıldız Technical University for 3 years. She is also the Vice President of Department of Biomedical Engineering of the Faculty of Electrical and Electronics of the same university. And now I turn the floor over to Görke Gürel Peközer.
Thank you for this nice introduction and inviting me to this Congress for presenting my research interests and recent work about vascularization strategies in bone tissue engineering. I would like to talk about tissue and organ losses and the bone defects, the place of bone defects in these tissue and organ losses. And as tissue engineers, what do we do about these bone defects, how to repair them by tissue engineering strategies? And I would like to focus on vascularization strategies in bone tissue engineering, because the main challenge of us in bone tissue engineering attempts is vascularization. So tissue and organ losses are a very common problem, which can be as a result of trauma, cancer, infection, or diseases. They can be lethal, so they can be very life-threatening sometimes, but it doesn't have to be lethal all the time or life-threatening all the time, because they considerably decrease the quality of patients, making them disabled and affecting them mechanically, aesthetically, and psychologically. And still, the golden standard in the treatment of these tissue and organ losses is to transplant their biological equivalents, which is conventionally done by tissue and organ transplantation. These are the statistics of tissue and organ transplantation from United States, and you see that more than 109,000 people are waiting for an organ or tissue to be transplanted. So the organ waiting lists are very huge, and they are increasing every day. Actually, 17 people are added on those lists every day. And only there are around 40,000 transplant procedures performed in a year. And sadly, actually, every 9 minutes, a person is added to these transplant lists. So it's a very huge burden on the society and also healthcare system. But being a donor, we can save more than 8 lives. So maybe it can be an awareness for you to be donors for tissue and organ transplantation. This is statistics from Turkey that I gathered from Statistics Institution of Turkey, but I could only had the 2017 data, because I think the Statistics Institution is dealing with measuring the inflation rate nowadays. But there are around 25,000 people waiting in the transplant lists, and only around 5,000 transplant procedures performed in a year. So many people, unfortunately, dying, waiting in the tissue and organ transplantation lists. And it's a very huge burden on our healthcare system, because even if they get the transplants from an appropriate donor, it's their rehabilitation and their pharmaceutical therapy is very expensive. And when they do not get any transplantation, again, their pharmaceutical therapy and rehabilitation is very huge burden to the healthcare system, and even more painful and hard for the patient himself or herself. And maybe, as I told you, it will be an awareness for tissue and organ transplantation and being a donor for tissue and organ transplantation. If you want to expand the organ donor pool, you can simply go to Enables and you can click that I want to be a donor, or it is that simple. And also, if you want to be a stem cell donor, you can have information on this web page of Türkök. They are very important to be an organ donor, and you can save many lives by being an organ donor. Bone defects are also represent a very big place in the tissue and organ losses. Actually, it is one of the most common tissue losses and represents a major concern as the life expectancy increases. More than half of all chronic disorders experienced by older people older than 50 years of age in developed countries are bone and joint disorders, and they are the most common cause of intense long-term pain and disability, decreasing life quality. I think you are already experiencing it with your elderly people in your family. They always complain about knee pain, back pain, etc. So it's very common in elderly patients. And this is becoming a very important socioeconomic burden to the healthcare system because our world population ages in time. And maybe it is reflecting the importance of bone defects in all defects of tissues and organ losses, because bone is the second most transplanted tissue in the world after blood. Of course, blood transfusions are the most common ones, but bone is coming after the blood transfusions. Although bone has an inherent ability to heal without leaving a scar. Normally, we observe this when we have a broken bone, then with an external fixation, the bone heals without leaving a scar. However, we have a concept called critical size defects, which we define as the smallest size of tissue defect that will not completely heal over the lifetime of a person or animal. These kind of defects cannot heal without a surgical intervention. So even until the end of the lifetime of a person or animal, the critical size defects will not heal, and these will be a huge burden on the patient, because they will cause a disability, pain, etc. until the end of the lifetime of that person. These bone defects can arise from infection, traumatic injury like a big car accident, tumor resection, congenital malformations and diseases, and they require reconstruction surgeries with bone grafting from the same person, from another part of the body, or from another person, from a donor. And also prostheses are one of the alternatives as the conventional methods for the treatment. The prosthesis we can define as an artificial body part to replace or augment a missing or impaired part of the body. They can be made of metals, ceramics, or polymers, or composites of metals, ceramics, and polymers, like, for example, metals and ceramics and polymers, etc. They have generally permanent, but they have an expiration date. They integrate poorly with the tissue at the defect site, and they can fail due to infection or fatigue. So these advantages drive the scientists to search for a better solution, because the problems with the waiting list for the organ transplantation and donor shortage related with the organ and tissue transplantation, and the problems with the prosthesis, like satisfying the patient's mechanical, psychological, and aesthetic needs, drive the scientists to search for a superior solution, because, of course, so many scientists also searching a mechanical way of compensating the tissues and organ losses. However, nobody wants a robotic hand. Maybe a few people can want, but always a biological equivalent is required for mechanical, psychological, and aesthetic needs of the patients. So tissue engineering is searching for those kind of biological equivalents. It is defined as an interdisciplinary field that applies the principles of engineering and life sciences towards the development of biological substitutes that restore, maintain, or improve tissue function. This is actually a definition by Langer and Vacancy in 1993, which is still valid today. Langer and Vacancy are the pioneers of the field. Actually, the tissue engineering research began as a response to the growing need for tissues and organs for transplantation, and it offers a lifelong solution and a better quality of patients' life, reducing the need for repeated hospitalization and healthcare costs associated with the pharmaceutical therapy. And we hope that it would be a better solution compared to the conventional treatment methods of tissue and organ losses. In a typical tissue engineering strategy, we employ 3 different components. One of them is the use of scaffolds. They can be made of natural polymers or synthetic polymers, biomaterials actually, which are very biocompatible with the body. We use cells. They can be autologous primary cells from the same patient. They can be allogenic primary cells from another donor, but the main strategy actually involves the use of stem cells and to differentiate them to the intended tissue. And another component of tissue engineering is the use of bioactive factors like cytokines, hormones, and small molecules that can induce a differentiation pathway, a vasculogenesis pathway, etc. In a typical tissue engineering methodology, the cells are isolated from the patient, preferably from the same donor, from the patient himself or herself, and the cells are isolated, then expanded in cell culture using the cell culture techniques, and then the cells are seeded on 3-dimensional scaffolds, and the scaffolding methods and the biocompatibility, pore size, porosity, and of course mechanical integrity and mechanical support of these tissue engineering scaffolds are important, especially for bone tissue engineering, because bone is a very is a tissue that loads very high loads. So the mechanical integrity and mechanical strength of the tissue engineering scaffolds are very important. Then after seeding the cells on these 3-dimensional scaffolds, then the growth factors are added, and then the cells are differentiating and beginning to show the tissue characteristics, and then these cells and the scaffolds are implanted to the patient's body, where the cells will regenerate the tissue and the scaffold will serve as a guide to guide the cells to their 3-dimensional architecture of the tissue and providing mechanical support until the tissue regenerates. The scaffold is maybe the most important part of tissue engineering strategies, because we know that simple cell culture techniques cannot be used to grow organized tissues, because in cell culture we can only obtain cells on monolayer. Okay, they cannot develop themselves into a 3-dimensional structure of a bone. So we need the cells need some mechanical, electrical, structural, and chemical cues. So we use some 3-dimensional scaffolds that we seed the cells on, and we also add some signaling molecules. And after some time in cell culture, the cells begin to differentiate towards the intended tissue. They, for example, if the aim is to engineer a bone tissue, the cells will mineralize, the cells will show osteoblastic phenotype, etc. And then the scaffold, the cells and the scaffold, this tissue engineering construct is implanted to the tissue, where the cells will regenerate a healthy bone, which is very similar to the native bone. So in tissue engineering strategies, the cell scaffold interactions are very important. After seeding the cells onto the scaffolds, you need to quantify the cell adherence, cell proliferation on the scaffolds, as well as the synthesis, as well as the secretion of extracellular matrix on the scaffolds, because the secretion of extracellular matrix will be very important, especially for bone tissue engineering. A mineralized matrix is the one that bears all the loads of the body that our native bones are taking. So the cytocompatibility, cell adherence, cell proliferation, and also cell differentiation on the scaffolds are the important points that we take into consideration in tissue engineering strategies. But normally, bone tissue engineering attempts at least, they're very successful. We can, even in cell culture, we can obtain a mineralized tissue which resembles bone a lot, which secretes bone extracellular matrix, which secretes bone proteins, etc. But the main challenge in bone tissue engineering strategies is the vascularization. Because bone is a very vascularized tissue. And when you consider bone, it doesn't like a vascularized tissue when you look at it macroscopically. It's like a hard tissue with made of only minerals, but of course there are cells, lots of cells in the bone, and the bone is traveled with lots of blood vessels and nerves. So we need to somehow vascularize the tissue because the cells in the tissue require oxygen and nutrients, and of course the hormones and inflammatory signals and cells come to the defect area in order to regenerate the tissue. Without a proper blood supply, we cannot have those. So generally grafts, bone grafts and tissue engineered constructs generally fail due to insufficient vascularization upon implantation, unfortunately. So the basic tissue engineering attempts generally fail to vascularize. So it is a very important challenge that we should do before implantation of these kind of bone tissue engineering constructs to the body. There are mainly three types of vascularization strategies that scientists are employing in order to obtain a fully functional vascularized bone construct. One of them is to implant the scaffolds in a highly vascularized region in the body, so that when the scaffold or the construct is first implanted into the body in a very highly vascularized region, then the vessels will invade the scaffold, and there will be a vascular supply inside the scaffold, and then the implant or tissue engineering construct is implanted into the right side, the defect site. Another strategy that is used to induce vascular endothelial growth factor secretion, which is very important for vascularization, and to induce endothelial cell migration in response to vascular endothelial growth factor. This is a strategy that we employed, and I will show you one example of it in my recent work. And another method to vascularize the tissue engineering constructs is to use a co-culture of endothelial cells with bone-forming cells. So it has lots of actually advantages in terms of inducing vascularization and also inducing osteogenesis for some reason I will talk about, because one of the techniques that I employ in my studies is to use of co-culture of endothelial cells with mesenchymal stem cells that will differentiate into bone cells. Since bone is a tissue formed by several cell types, co-cultures of heterogeneous cells can better mimic the in vivo microenvironment than monotypic cell cultures. So co-culture does not only induce vascularization, but also induces osteogenesis in some way, because the bone is a very vascularized tissue, and the bone cells are very in contact with endothelial cells in the bone, which make the cell-to-cell contact between bone cells and the endothelial cells particularly important for bone tissue. Besides, co-culture of endothelial cells with bone-forming cells allow researchers to study how the cellular crosstalk between these cell types affect functionality of the others, because interactions between heterogeneous cells provide the cues essential for differentiation, organization, and homeostasis as soluble signals and via cell-to-cell interactions. So there are two main strategies that employ co-culture techniques. In first one, we use direct cell-to-cell contact, and we culture the bone-forming cells and endothelial cells together in the same place so that they can make cell-to-cell contact, and they can share their cytokines or molecules with these direct cell-to-cell contacts. However, also using co-culture techniques with cell culture inserts, etc., are also one type of doing co-culture. I will be talking about those two ones in this talk. One of the studies that I have done, actually, the work is published in 2016. It is the influence of co-culture on osteogenesis and angiogenesis of bone marrow, mesenchymal stem cells, and aortic endothelial cells. So in this study, we isolated red bone marrow stem cells, mesenchymal stem cells, from the bone marrows of rats, and also we have isolated endothelial cells from aorta of rat, and we co-cultured them directly in cell culture, and we examined the vascularization and osteogenesis. And we wanted to observe how the vascularization and also osteogenesis is affected by this co-culture, because from the previous studies, we know that the osteogenesis is also affected positively by the co-culture of mesenchymal stem cells and endothelial cells. So we examined the vascularization after 14 days and 21 days of co-culture under confocal microscopy, and we also quantified the tube length from the tube lengths of the tubes that are formed in the cultures, and the number of branching points, which are very important in a vascularized tissue. And we have seen that when we co-cultured them, both the tube length and the branching points are superior compared to the bone marrow stem cells or endothelial cells alone. And we have identified lots of microvessels or structures that resemble microvessels, and lots of branching points of the microvessels when we co-culture endothelial cells with mesenchymal stem cells compared to the mesenchymal stem cell culture alone and endothelial cell culture alone. You see that the tissue formation, the microvessel formation are superior in the co-culture cultures. We also looked at the mineralization, which is very important for bone tissue because bone extracellular matrix is composed of primarily collagen type 1 and also calcium phosphate minerals. And the minerals are the ones that they take the compressive loads of the bone, of the skeleton on themselves. So the mineralization of the cultures are very important. And we observed that after 21 days, the mineralization is very intense in the co-culture environment. This is surprising because when we cultured mesenchymal stem cells alone, we couldn't observe that much of mineralization. However, when we add endothelial cells to the culture, then the mineralization is increased. This is especially important because I think in normal native bone, mesenchymal stem cells and endothelial cells are in the same environment, and when the oxygenation of the environment increases, then the bone differentiation and mineralization also increases. We also looked at ALP activity of the co-cultures, bone marrow stem cells and endothelial cells alone. ALP is an important enzyme that shows osteogenesis to the scientists, because it's actually alkaline phosphatase enzyme which cleaves phosphate from phosphate compounds, so that the bone cells can use that phosphate in the structure of calcium phosphate minerals of the bone minerals. So the bone cells are using this phosphate for making bone minerals. Okay, so alkaline phosphatase activity is increased in cells that can differentiate into bone. And when we examine the ALP activity in 3 different groups of co-culture, mesenchymal stem cells, and endothelial cells, obviously endothelial cells do not have any ALP activity as expected. However, the co-cultures of endothelial cells and mesenchymal stem cells have increased ALP activity. And when we look at the co-cultures that we did not use any differentiation factor, we actually observed a high ALP activity. Because normally in tissue engineering strategies, when we culture stem cells in the cell culture, we feed them with differentiation factors like glycerophosphate, ascorbic acid, dexamethasone, etc., so that we can drive the stem cells into bone cells. However, in this one, we didn't use any differentiation factor, but only adding endothelial cells to the medium, the cells could differentiate into bone. Because for mesenchymal stem cells, there are actually 3 fates that they can choose from. They can be a cartilage cell, a bone cell, or adipose cell. And if there are some endothelial cells in the culture, they prefer to differentiate into bone cells, because in the native bone, they're in contact with each other, bone cells and the endothelial cells. So even without the addition of any differentiation factors, the co-cultures could show intense ALP activity compared to mesenchymal stem cells with differentiation factors. Okay, but if you compare these 2 groups, in which one of them differentiation factors are added and the other one there is no differentiation factors, when the red bone marrow mesenchymal stem cells are cultured alone, the ALP activity is higher than the mesenchymal stem cells with no differentiation factors. However, in co-culture, the addition of endothelial cells made a huge difference in terms of ALP activity of the cells. We also checked for the osteogenic genes by real-time PCR experiments after 14 days and 28 days of incubation in cell culture. And we have seen that after 14 days of culture in co-culture groups, we observed an increased osteocalcin, osteonectin, and RNX2 proteins. Osteonectin is a very important extracellular matrix protein of bone, and RNX2 is actually a very important key transcription factor which shows the commitment of stem cells into osteoblastic lineage. But after 28 days, we saw a big difference in terms of osteogenic genes expressed from co-culture groups. Both osteonectin, collagen type 1, RNX2, and osteocalcin expressions were higher in co-culture groups. So this study showed us that the interactions with endothelial cells and mesenchymal stem cells draw the mesenchymal stem cells into more of an osteoblastic phenotype, and we can use this for vascularization, of course, endothelial cells should be there for vascularization, but also osteogenesis is positively impacted by the presence of endothelial cells. In another more recent study that is titled as Investigation of Vasculogenesis Inducing Biphasic Scaffolds for Bone Tissue Engineering, we employed a different method. In this time, we did not want a direct contact between the endothelial cells and the mesenchymal stem cells. So we used an in vitro cell culture model, but we also engineered a biphasic scaffold for bone tissue engineering that will induce vasculogenesis. The scaffold was made of wet spun PLGA. So PLGA, polylactic acid glycolic acid, is a polymer which is a very biocompatible polymer. We made a fibrous scaffold made of PLGA using wet spinning technique that you will see here, and then we covered it with a PLGA PEG PLGA hydrogel, which is again a biodegradable hydrogel, and we embedded GS4012 small molecule, which is an inducer of vascular endothelial growth factor, into this hydrogel, and then we seeded the wet spinning scaffold with red bone marrow stem cells, which are stained with a quantum dot, which stains them into a red color. Okay, so the mesenchymal stem cells are red, that you will see in later slides. So there were GS4012 in the polymers, in the hydrogel, and then we intended these mesenchymal stem cells to take this GS4012 and induce vascular endothelial growth factor secretion. And as a response to vascular endothelial growth factor, the endothelial cells that are green, that are stained with green quantum dots and stained in the upper chamber of this cell culture insert, can migrate from the pores of the insert onto the scaffolds, okay? So that they will form a vascularized network, and also there will be a direct contact between the mesenchymal stem cells and endothelial cells, which can drive the mesenchymal stem cells into a more osteoblastic phenotype, and also endothelial cells will form a proper vascular network. We designed this in vitro model because actually we wanted to test also this scaffold, the vascular endothelial growth factor inducing scaffold and vasculogenesis inducing scaffold in red calvaria. But we did not use any endothelial cells for this in vivo studies, so that we wanted our scaffold to induce endothelial cell migration from neighboring tissues. Okay, so we measured the GS4012 release from PLGA PEG PLGA hydrogel, and we obtained a sustained release into the cell culture media from the PLGA PEG PLGA hydrogel, and we looked for endothelial migration by confocal microscopy. So we wanted to see if the endothelial cells, which are stained in green, migrated onto this scaffold, biphasic scaffold. Okay, and the main aim is to secrete vascular endothelial growth factor from mesenchymal stem cells in response to the GS4012 in the environment and the endothelial cell migration towards this scaffold. And actually, we have seen lots of endothelial cells migrated to the wet span scaffolds. And this migration was comparable with the vascular endothelial growth factor added group. So in some of the groups, we added vascular endothelial growth factor to the hydrogels. However, we did not prefer to add vascular endothelial growth factor as our experimental studies, because it has a very short half-life and also use of growth factors is an expensive method for tissue engineering strategies, but GS4012 is a small molecule which has longer half-life and also cheaper than VEGF, and using it will be very beneficial for maybe for the marketing steps of this biphasic scaffold. So we have obtained endothelial migration compared to a very good endothelial migration compared with the actual vascular endothelial growth factor added group. And we also looked for mineralization of mesenchymal stem cells, and we obtained a good mineralization at day 20 in cell culture. And these are the wet span scaffolds, and the cells are seated on these wet span scaffolds, and we observed very good mineralization after 21 days of incubation, and the cells were secreting so many minerals. And we performed scanning electron micrography of cell-seeded wet span scaffolds, and we saw that cells could adhere to our wet span scaffolds, and they can proliferate, actually populated and covered all over the surface of the wet span scaffolds, which shows us that the wet span scaffolds are cytocompatible and can be applied for tissue engineering strategies. We also quantified the bone and endothelial specific genes, as well as VEGF secretion from cultures by ELISA assay. So when we cultured the cells with GS4012, we have seen that an increased VEGF secretion by mesenchymal stem cells up to fourfold compared to the group that is not added with GS4012. And we have seen that endothelial migration was up to threefold compared with the group that is not added with GS4012, but the endothelial migration was decreasing in time because more and more endothelial cells were migrating towards our scaffold, but the endothelial cells seated on the upper chamber is limited, so the amount of endothelial cells were decreasing, the migrating endothelial cells. We have up to 30-fold ALP activity with the groups that we added GS4012. So somehow the endothelial migration and the presence of vascular endothelial growth factor is increasing the ALP activities and the expression of ALP gene. And also we saw up to twofold of RENX2 expression, which is, as I told you, a key transcription factor in osteogenesis, and the RENX2 expression was high in both 7 days, 14 and 21 days of incubation in cell culture media in the presence of GS4012. And we have a good expression of osteocalcin and also collagen type 1, which are very important proteins in the bone extracellular matrix. So if we want to direct the mesenchymal stem cells into bone-forming cells, osteoblasts, osteocytes, etc., we need to quantify the expression of bone-related genes. Of course, another aim is to migrate for the endothelial cells to migrate onto our scaffold. So endothelial specific gene expression is also very important to us. And also we quantified VEGF secretion from cultures by ELISA assay, and we have seen a very good VEGF secretion after 21 days of incubation with GS4012. Actually, we have tested GS4012, which is known as VEGF inducer, but we want to see if it will induce the VEGF secretion from our endothelial cells, red aortic endothelial cells, and we have seen that when we add GS4012 to red aortic endothelial cells, they increase their VEGF secretion up to 3-fold. So it's a very good small molecule for inducing vascularization in tissue engineering constructs. So we aim to use this GS4012 small molecule, which is not used very commonly in the literature to induce vascularization. And also we are in search for other type of small molecules that can induce vascularization and other vascularization methods that can be employed for bone tissue engineering attempts. So we also performed in vivo bone formation on the red calvaria. For this one, we made 88 millimeter calvarial defects on the red calvaria, and we put the wet span scaffolds and the cells, and then we put some PLGA PEG PLGA hydrogel with GS4012, and then we covered the surface and waited for 4 weeks and 8 weeks for bone formation. And we employed histomorphometrical analysis for the recovered bone after 4 and 8 weeks, and we have seen increased vascularization and also bone formation in the groups that we use GS4012 and mesenchymal stem cells, as well as the wet span scaffolds and also PLGA PEG PLGA hydrogel, which shows us that actually in vivo we could obtain a nice bone with vasculature that will benefit us a lot, because the main problem in bone tissue engineering attempts is the vascularization. However, with the use of GS4012, we could increase the VEGF secretion from red bone marrow stem cells, and as a response, endothelial cells could migrate from neighboring tissues, because in the in vivo analysis, we didn't use any endothelial cells. Endothelial cells should migrate from the environment. That is a process which we see in a native original tissue healing process in bone. So the endothelial cells are migrating from the neighboring tissues, from the healthy bone in the neighborhood, and then they are colonizing there. They are forming microvessels, tubes, etc. And we have seen that tube formation in the histology, and also we histomorphometrically we quantified bone formation and also microvessel formation in these groups, and we have observed superior bone formation and vascularization in the groups that we used GS4012, which is a small molecule that induces VEGF secretion. So this is all from me. Thank you for listening, and I will wait for if you have any questions regarding my presentation.
Thank you for the information you provided. Now here are the questions posed to you.
Yeah.
The first question, what is the latest development in tissue engineering, especially in osteogenesis applications?
Actually, as I am searching about new literature, recent literature from the databases, I see that most of the scientists are dealing with these vascularization methods, because as I told you, to make a bone, even in vitro conditions, is not a very hard issue. We can do it. We can have a very good mineralized bone tissue. However, when we implant those tissue engineering constructs to the patients or to the animal models, the tissue engineering constructs are failing, because then the blood supply is not enough for them. And without any blood supply, the interiors of the tissue engineering constructs are going into necrosis, they're dying, and eventually the tissue engineering attempts will fail. Okay, so the recent advancements generally are done in the vascularization field.
Second question, what do you think of using 3D organoid systems instead of co-culture systems?
Yeah, they're also very important, and we are trying to deal with them nowadays, actually. Of course, in order to study co-cultures, in order to test them without any animal models, without any animal sacrifice, will be very important for us, and we are dealing with them.
Last question. I've seen your work in research, investigation of vasculogenesis, including biphasing scaffolds for bone tissue engineering. What is the main difference between these two-phase studies?
Two-phase studies, biphasic or monophasic scaffolds, I think it is asked. Okay, so we have used biphasic scaffolds. The first phase of the scaffold is made of PLGA, wet span PLGA, which serves as an actual tissue engineering scaffold to guide the cells and to give mechanical support to the tissues. But the other phase is PLGA PEG PLGA hydrogel, which is doing the GS4012 release. So it's like a drug delivery phase, and the other phase is wet span PLGA scaffold, which is providing mechanical support and guiding the cells into three-dimensional structure of the bone tissue.
Okay, thank you very much for participating. It was a very productive conversation.
Thank you. Also, I would like to thank you to the organizing committee, to Esra Hoca for inviting me here, and also to you, of course, for listening to me in this sunny weather on a Saturday. I hope it will be a productive congress for all of us. Thank you again.
Görki Hocam, teşekkür ederiz.
Ben de teşekkür ederim Esra'cığım.
Seni görmek çok güzeldi. Bir sonraki
Aynı
aktivitede gene görüşmek üzere.
İnşallah. Çok teşekkür ederim davet ettiğiniz için her zaman.
Cumartesinin geri kalan kısmını bari güzel geçir.
Eminim sizinki de güzel geçecektir. Görüşmek üzere. Hoşçakalın.
Görüşmek üzere.
Kongreler diliyorum.
Teşekkür ederiz.
Now break, and then Yavuz Nuri Ertaş will be with us.
Professor. Our scheduled guest, unfortunately had a medical emergency and she can't present for us. Unfortunately, we hope she gets better soon. Therefore, we are moving along to invite Professor Yavuz Ertaş from Agios University to present to us regenerative medicine and biomedical application. Welcome, Professor.
Thank you for the introduction. Is my screen visible?
No. Yeah, now it's okay. It's visible.
Okay. So I will present you a research related to radiotherapy. So how can we use nanoparticles for radiotherapy? But before that, I want to introduce myself and a few research directions, information regarding our lab, and then I will switch to the main topic. So here I work at Erciyes University, and before the Erciyes University, my undergraduate education was from Başkent University, Biomedical Engineering. So as an undergrad, I was educated as an engineer in biomedical engineering field, and then after that, I went to master's to Bilkent University in Material Science and Nanotechnology program, and after graduation for PhD and postdoc, I was at UCLA. And early 2020, I returned to Erciyes University through the TÜBİTAK program called 2232. And then where I started, and for the last 2 years, we set up our lab and started our different research directions. So I wanted to introduce this slide. This is a work that I was involved in when I was as a postdoc at UCLA. So here, the title of the talk was Regenerative Medicine. And here, and the previous speaker mentioned that vascularization is the important issue in these scaffolds. Actually, this is exactly what it's related about. So let's say you have a heart attack, and what happens is then the heart cells die. So if you want to regenerate that tissue, you either need to externally inject the heart cells, or you have to make the adjacent tissue to regenerate in that area. So there is a field called 3D bioprinting scaffolds. So as you see in this picture, the yellow mesh. So what you do is you print heart cells with another carrying polymer or material in a scaffold manner, and then you implant the scaffold into the patient, and you expect the vascularization and the tissue regeneration in the area. But one of the big problems is that the cells need nutrition and oxygen to survive. And in the area where the tissue is already dead, there's not enough vascularity. So even the cells you implant in the scaffold, before they can generate a tissue, they generally die because of the fact that they cannot reach the oxygen, because you cannot provide oxygen. So in this study, what we did was we mixed the scaffold material, the cells, heart cells, with a material called calcium peroxide microparticles. The calcium peroxide is a material which, upon contact with water, releases oxygen. So, and then we showed that it can before the vascularization can start, which is around like a week in vivo, these microparticles can give enough oxygen to these heart cells in the scaffold to regenerate the tissue. And this was published in a prestigious journal. So I want to first talk about the ongoing projects and what we do, and then the specific topic of today. So right now, I am working as a PI in 2 different projects, and the first one I mentioned, the 2232 project. In this project, we are trying to produce highly magnetic nanoparticles using some microfabrication techniques. And in the other project, which has recently started, where we are working with a team of researchers and trying to develop some treatment for the COVID-19. And besides this, we apply to several other funding agencies for support. And as you may know, most of them are also getting rejected. This is the reality of the grant applications. But we applied through a consortium. We applied a postdoctoral program scheme where the title is the Interdisciplinary Solutions in Bioengineering and Nanotechnology, and it's supported by the Horizon. So we have partners in this program, but we will hear about the results in the late summer, I guess. So these are the partners. And then I also try to attract like talents abroad to Turkey, to especially my institution. So through the program where I also came back to Turkey, the 2232B program, the Young Researcher program, Dr. Syed Mir Sayed this year received this grant and he will join RGS University. And through another program called Co-Circulation, it's 2232, and Dr. Mustafa Abait from Italy will join us. And I kind of serve as like a mentor or the consultant in these projects. So in a few slides, I want to just give a broad view on what kind of topics we work on, and then I will switch to the main talk. So the first title, as you know, this is the project. I returned back to Turkey, the 2230 project. We are trying to produce highly magnetic nanoparticles. They look like different shapes, like nanoball shapes. And these different shape, it gives different magnetic properties, and we are trying to enhance the magnetism of these particles. And besides these, we're trying to produce nanofiber scaffolds, and we try to load drugs and nanoparticles in these. And what happens is that once you shine near infrared light to these scaffolds, the nanoparticles, the metal nanoparticles absorb this light and heat up. So once they heat up, they started to kill the cells around them. So let's say in a tumor model, if you implant these and if you apply light from outside, it can heat up the particles and it can kill the cells. In this case, it's cancer cells. But one limitation of this technique is the near infrared light. The wavelengths are around 750 to 1000 nanometers roughly. Has a like shallow penetration to the human skin or to skin. So what happens is that the other molecules or materials absorb or scatter the light. So, but in some cases where the cancer is close to the outside of the body, like to the skin, you can use this kind of photothermal therapies. So another research area we're trying to work is, in nature we have like different insects. Cicada, and in these insects, on their wings, they have these kind of like structures. So where these structures are antibacterial. So the way it works is kind of running in this video. So when the bacteria sits on this surface, it experiences a stress. So its membrane ruptures and cytoplasm is just damaged. So the bacteria dies. So this is how some of these insects are fighting with the bacteria. So one big field of research is producing antibacterial surfaces. This is like this is a long-standing topic. Tens of years has been spent on this topic because implants, let's say in medical implants, even if you have a great implant that does the job, it can carry some bacteria, and bacteria can grow on that, and it can generate some other problems. In this case, for example, inside the ear, there is some bacterial infection. So what if we produce surfaces antibacterial, and in the long term, we apply these techniques to implant surfaces and make them also antibacterial as much as possible, and then achieve healthier and safer materials. So this was another topic, and I will present a few more here in the slide, and then I will start the talk. So upconversion nanoparticles, again, these are particles. They absorb the infrared light and emit UV light. UV light can also kill the cancer cells, and also metal nanoheterostructures for in vivo radiotherapy. Actually, I will talk about this today. So what happens here is that radiotherapy is a therapy method where X-rays are used, and metal nanoparticles can absorb these and release electrons around them, which also can kill the cancer cells, which I will go into much more detail in starting the next slide. And we are also working on 3D printed scaffolds towards bone regeneration. What happens is that you can mix nanoparticles, whatever nanoparticle you're working on, with different scaffolds and implant them in a mouse model. And then you apply either light or X-ray or magnetic hyperthermia or other chemotherapy kind of effects to make sure the drug inside the scaffold or the nanoparticle does its job, either heating and killing the cells or releasing the drug in a slowly controlled fashion to kill the cells, cancer cells, or some other effects can also be observed. So, one last slide. Actually, this is a topic that we are trying to understand and also find solutions. So the professors at the dentist faculty at RGS, said that there's a problem called dentin hypersensitivity. This is what here is happening is that in dentin, there are these tubules, microtubules, going inside to the center of the teeth. And if your teeth is kind of has a thin dentin layer, or in some cases no dentin close to the part where it touches the like meaty part, and these tubules are exposed to the outside, and there is a liquid inside these tubules, and once you drink cold, or you in cold weathers, or sometimes this fluid inside these tubules can move around, which generates pain. So, for example, in these cases, when you do this, so that air goes inside these tubules, and the water, the solution, the liquid inside these tubules moves around and creates pain. So one problem is that the dentists are asking, can you please seal this area, block this entrance of the tubules, so that the liquid inside doesn't move and the pain is avoided. So actually there's commercially available glues, but the problem is they are effective only just a few months. After a few months, they either fall out, because teeth is a part where there are a lot of force where it experiences a lot of force through chewing or other actions of movement. So ideally, you want something that closes these tubules and reduces the pain. So now I will start the main part of the topic. As you can track, I have the slide number here. I have 49 slides, and it will finish on time. So in a cancer treatment approach, we have a few different modalities or approaches. One is the surgery, and the other one is the chemotherapy, and the other one is radiotherapy. So if you have the cancer, these are the most common approaches right now in today's clinics that will be used. So what is radiation therapy? And even after surgery, let's say the surgeon removed the tumor tissue, but also there is always, or most of the time, a bit of like a few cells or some invisible chunk of tumor tissue left behind, and it can regrow after some time. And in a 6 months or 1 year or a few years, it can regenerate the tumor again. So ideally, you want to kill as much tumor cells as possible after the surgery, using either chemotherapy or radiotherapy. So radiation therapy, that's what the topic is today. What happens is that you send X-rays to the tumor. The patient lies down on a machine, inside a machine, and X-rays are sent. And when X-ray touches a tumor tissue, what happens? It can kill the cell through two different effects. One of them is called direct effect, and the other one is the indirect effect. In the direct effect, X-ray directly damages the DNA of the cell, or it can create mutations. Or in indirect effect, it can touch the water of the cell, and the water can generate radicals or hydrogen peroxide, which can also indirectly attack the DNA and kill the DNA. So once the DNA is killed, the cell dies. So this is how the X-ray is used. But the problem here is X-ray, until it goes to the area you want to focus X-ray, or you want that to go, it goes through the healthy tissues. It goes through the skin and whatever is on the way. So while it is going to the specific location inside the body, it is also harming the healthy tissue. So ideally, you want some material there, and ideally you want to reduce the dose of X-ray as much as possible. So these are called radiosynthesizers. So radiosynthesizers are generally metal nanoparticles where they are high atomic number. So what happens is that when an X-ray hits them, there are different kinds of electrons scattered or emitted from them. So these emitted electrons can kill the DNA much easily. And the higher the atomic number of the particles, the better the treatment efficiency. So here, as I said, extracted electrons can either generate reactive oxygen species and kill the DNA, or they can generate radicals, hydrogen peroxide, or other agents that can also kill the DNA. But the function is through these electrons that are scattered, that are emitted and scattered. But that is also caused by the incoming X-ray. So here, how can we connect the nanotechnology with this problem? So here, you want to reduce the dose of X-ray. You want to focus that in a very small volume. So what happens is that if you have very small particles in that area specifically, you can send just a little dose and you can generate the same effect or maybe more than the previous effect. So in this case, nanoparticles are emerged as a possible alternatives. We have to understand how nanoparticles should go to the area that you want to do X-ray. You want to do the radiotherapy. So in healthy tissue, the distance between the blood vessel cells are kind of narrow. And there is also a lymphatic system that can drain the foreign objects. But in tumor site, the distance between these cells increases. So the nanoparticles can easily go inside these, go through these distances. And the lymphatic system in tumor cases, in cancer cases, is weak or missing, so that once it goes inside that, once nanoparticles goes inside the tumor, they stay there. They cannot be easily removed. And this is called EPR effect, but EPR effect is not the only choice and ideal. So there is, and this is called passive targeting. You just inject the nanoparticles through the blood vessel, and they go to the tumor site to some extent. And yeah, that's what I think I want to mention another thing here. So there is also this and another illustration that the distances between the cells are high here. So, but the thing is, we should actually, we shouldn't rely on the passive targeting only, because passive targeting, the delivery efficiency of nanoparticles to the desired site is not very high. So what happens is that if your tumor cell, and not if, in general, tumor cells have specific receptors around them, depends on what kind of tumor is that or cancer is that. So let's say most of them has a receptor called folate receptor. So if you decorate your nanoparticle with some targeting agent which recognizes this folate receptor and binds to it, so you can kind of deliver your particles to the desired site in a more controlled and higher efficient way. So this is called active targeting. And active targeting is done through different surface modifications. And I will not go into that much detail. So I will start now, after this brief introduction, I will mention what we have done experimentally and what are we working on, and why are we working on these, and what are the results, what are the future directions. So as I said, high atomic number elements are desired. So in this case, we designed gold and gadolinium based nano heterostructure, which means in this case the nanoparticle has two components. One of them is the gadolinium, the other one is the gold. And actually there are some other details that when gold and gadolinium is in touch, there's some electron cloud overlap, and there's more enhanced electron scattering. But what happens is that this is bovine serum albumin. We kind of functionalize the particle surfaces with some protein, and then we produce them. The first step is the production of the nanoparticle, and then you have to verify if the produced particles in terms of size and the charge and material component are what you want. So in this case, we have the TM image here, and they look dispersed. This is the first step of confirmation. It gives a good idea that they are kind of uniform shape and they are well dispersed. And then you have to check step by step if your synthesis is working. So let's say in this case, we only have the gadolinium oxide and BSA. The image resolution is a bit low, but as you can see, there are like particles here, right? And then once we add the gold, as you can see, there are like dark and light areas. So actually, this is the heterostructure itself. You know, gadolinium atomic weight is higher than gold. So the gadolinium looks more darker and the gold looks more white. And the closer look up, more zoomed in vision, as you see, it's clearly there are like some different areas than the other part of the particle. So this is a heterostructure composed of two different metals. So we also need to verify this through TMEDS, so elemental mapping. So this is like the gadolinium. I think that it's not clear here. This is gold, and this is the I don't remember this. This is the merged image. So you have to verify the elemental. So in this, for example, in this image, you see there are two different things, but you don't know what they are, right? So you need to make sure that one of them is gadolinium, the other one is gold. So that's why you do elemental mapping to make sure there is the desired components, the elements inside the particle. So then you go to other characterization techniques to further verify the proper synthesis of the particles through XRD or FTIR UVVs. I will not go into very much detail, but all these analyses are required if we want to make sure the particles are first as what we aimed for. And after we get the initial characterization done, when we consider injecting these particles in a mice, let's say in a mice tumor model, we need to make sure these particles will not create adverse effect in the bloodstream. So which means there's a test called Hemolysis Assay. So here, the red blood cells, and normally in water, their blood cells are destroyed and the hemoglobin inside comes out. They look like red in color. And in PBS or in some control solution, they should look like colorless. So what happens is that you have to produce different concentrations of nanoparticles, and you have to do this test so that you should see if my nanoparticles are destroying or kind of rupturing the red blood cells or not. So there is a standard limitation saying that 5% is the maximum level that can be tolerated. If it's more than 5%, you cannot use that particle. You cannot inject that in a human or mice or animal, any other animal model. So as the concentration goes up in these two cases, as you see, the difference is that here we have gold. The hemolytic activity rises as it makes sense because you have more particles, but it's all well below the threshold level. So you, after confirming the first compatibility through the hemolysis, we also move on to the in vitro test. So what is in vitro test? We want to make sure if these particles are killing the tumor cells, which is 41 cells are the mice tumor cells, in an in vitro way, not in the animal, inside the animal, outside the animal, and after and before X-ray. So what happens is that there's an assay called live dead assay, calcium AMPI. So here there's a dye. This is a dye. Normally it's colorless, but once it goes inside the cell, it has some reaction with the esterase of the cell, and it shines green. So once it shines green, we can say that the cell is alive. And on the other side, there's another dye called propium iodide. It's PI. It binds to DNA, and once this dye is inside the cell, if it binds to DNA, it's red. Normally, it shouldn't bind to DNA because DNA is inside the nucleus. But if the cell is dead, the DNA can be anywhere around inside the cytoplasm, and the PI can bind it, and it can look like a red color. So green means the cell is alive, red means the cell is dead. So here we have the 4 groups for each calcium AMP and PI tests. So the left side is the control group. In the control group, we don't have any X-ray, we don't have any nanoparticle. Normally, the cells should grow and they all should look like healthy. As it's the case of calcium AM, we see the green color very heavily, and PI, we don't see any red. So which means the cells are all alive. And under X-ray, this is X-ray is the similar dose when you go, unfortunately, hopefully you don't not, but if you go to a clinics and if you get an X-ray dose, so this is the 4 grade, this is what it will look like. So which means, look, the level of green decreased and the red started to come up, which means cells are dying to some extent, not totally. After these particles with X-ray is used, look, there is no gold here. As you can see, the effect is increased from here to here. The green is lower, red is higher. But once we add the gold, as you can see, the effect is further increased. So which means not just the material, but also the material combinations, the nanoheterostructures are also very efficient in X-ray therapy. Why? Gadolinium atomic weight is normally, higher than gold. So there is ideally, the first thought is like, why would you add gold to gadolinium? Because it has already higher atomic weight. But once you have these heterostructures, the overlap of the electron clouds enables X-ray, enables the incoming X-ray to emit more electrons, so that it can do a higher effect. So this was the first level of confirmation. And then there is another assay called clonogenic assay. This is where you're checking if the cells can transfer the information they have to the further generations. What happens is that you dye the cells with a crystal violet dye, and it looks like this blue purple color. If the cells are alive, you see more purple or blue, and if the cells are dead, you see less. So as you see, the systematic investigation, it looks much less here as well. So the data from the in vitro calcium AM live dead cells and the clonogenic assays make sense, as also seen in this chart. And further, we want to make sure if these nanoparticles are effective and inhibiting the cell division. So for a particle to kill the tumor cell in a very efficient way, it has to catch the cycle of division in G2 and M phases. So there are like several different phases, but if the treatment arrests the cell division cycle in these two phases, it's more effective. And through flow cytometry analysis, we check just X-ray alone, X-ray with gadolinium oxide, X-ray with gadolinium oxide and gold heterostructures. These are the percentages of G2M cycle arresting overall in overall compared to all cycle numbers. So as you see, there is also increase in this trend as well. So addition of gold increases the arrest of the cell division cycle in G2 and M phases. And then after in vitro confirmation of the treatment efficacy, you have to, this is the end test. You have to inject these to an animal and see if they're working properly. So what happens is that you inject these nanoparticles to a mice subcutaneously, and then you put them under X-ray. First you wait until the breast cancer is developed. So you wait a few weeks, 2 weeks, and then you see there is a bump here. This is the breast cancer. You first generate breast cancer in an animal, and then you anesthetize the animals, and under an X-ray machine, radiotherapy machine, you send X-ray to the animals, and you then over the time check if the tumor volume is decreasing or increasing. This is how it looks like. And then what happens is that, again, we have the 5 groups here: the control, X-ray alone, X-ray gadolinium oxide with X-ray with gadolinium oxide and gold, and just the nanoparticles without an X-ray. So as you can see, the control group, the tumor volume is getting like 10 times almost in 3 weeks. In X-ray alone, which is right now happening in the clinics, it's still growing, but not as much as the control group. And as you see, as we go to red, the increasing tumor volume is decreased. One thing I should note here is that, as you see, there are standard deviations. In some cases, actually, tumor volume decreased. Actually, this study, there were 6 mice on this case, should be increased to further refine the time-dependent manner of this treatment. So this is another talk. This is another topic I want to mention. So you can use different metal nanoparticles, and you can test the efficacy for the radiotherapy again. So in this case, what we did is we have silver sulfate nanoparticles. Again, we have a heterostructure here, and then we coat them with folic acid. So folic acid, as I mentioned at the beginning of the talk, is a molecule that can target, folic acid can target the folate receptors of the tumor cell, right? So once folic acid and the receptor is in close proximity, nanoparticle can bind, and it can be internalized, and it can be uptaken by the cell. What we want is, we want the tumor cells to uptake these particles. So we put kind of like signal molecules around them, so the cancer cell thinks like it wants to like it inside it, it goes inside. So I'm not talking about much the synthesis and other characterization briefly. again, what we have is that we have a regular TEM image here, and we have STEM image, and we have an idea about what the size of the particles are, around like 30, 40 nanometers. But then we also check the hydrodynamic diameter. So hydrodynamic diameter is once the particles inside solution, it experiences a higher size effect. It can have a screening size, so it experiences a higher size. So before and after folic acid addition, as you see, the folic addition kind of more than doubled the size of the hydrodynamic size of the particles. And here through UV-Vis experiments, you confirm the peak of the folic acid around 400. So this is how you should proceed. You know, if you do not have the confirmation here at UV-Vis about the folic acid peak, then you go back to the synthesis and you say, probably I did something wrong. I should edit. I should change the way I do things. And then you go to hemolysis assay again. As I said, the 5% is the threshold, and in all concentration, it was less than that. And the cell viability for these particles, but this is healthy cell viability. You want to first check your particles are killing the healthy cells or not, because you don't want to kill the healthy cells. And in in low to moderate concentration, the cell viability is very high. As you increase, you know, it starts to kill the healthy cells as well, but this is kind of tolerable, and we don't need that much concentration in general. So, and then we need to check the cell uptake. You know, we want to make sure that cancer cells take these particles inside them. So for each concentration, we check what percentage of the particles in vitro are uptaken by the cells. As you can see, as the concentration increases, the uptake rate is also increasing. And for the folic acid here, important thing, after the addition of folic acid, as you see, the uptake in all cases increases. As it makes sense, the folic acid is the targeting receptor agent. So, and then we check the tumor cell viability for all these groups. So what do we have in these groups? The control group, there's nothing done. It's like 100% cell viability. The X-ray, the viability decreases to some extent. And then the just particles, you know, the particles does not have that much effect. I mean, if you go to very high concentration, then it might have some little effect because there's no X-ray here. And then in this case, we have the same particles as X-ray. As you see, in all subgroups, the cell viability decreases after the addition of X-ray. By the way, this is just one-time shot X-ray. If this is like one-time shot, and then after days, it was like evaluated. Normally, you can, if you do this every month, like 3 months, once a month, the effects are much higher. Here, what we are trying to show is that if there is also this much effect in just one shot, it can be very effective way of approaching solving these problems. So, and then addition of FA, you know, in this case, as compared to here, there's not much difference because there's no X-ray. And then here, maybe it's not clear to see, these levels are lower than without FA, because there is the cells uptake the particles with the folic acid more than the other ones that thought they don't have it. Again, we do the same clonogenic assay, and we see in this case the color is the least, which means it's the most effective treatment. And then we also check survival fraction, and this is the animal survival fraction. As you see, this has the most effect. And then we go to in vivo, the animal models. We inject these particles in mice, and then over time check their tumor volume and their body weight. Ideally, your treatment should not change the body weight that much, because if you are fixing something through creating some other problems or through making the body weight decrease a lot or increase a lot, then there is something else probably going wrong. So as you see, it's for all cases, it kind of looks stable, but for the tumor volume, you know, as you see, this group is decreasing or keeping the tumor volume constant compared to all other groups, which also confirms the approach is effective. So this is the last study I'll talk about. By the way, these studies are mostly under review or under consideration by journals, and that's why I share just most of the data, not all. So in this case, we have a different nanoparticle where we have the iron content and the bismuth content. So, you know, the iron here, actually we haven't done this, but iron here can also allow the nanoparticles to track using MRI. without in an invasive way. So once we have these particles, again, we synthesize them, the heterostructures, and then we do characterizations. Again, we have the heterostructure. Look at this particle. There is dark region and the lighter region. So there are two different materials. And then we do some other characterization. We try to confirm the existence of bismuth and iron, specifically, also BSA. And I will not go into that much detail. And then we do the hemolysis test again. Again, as you see, they're all below 5%. If it's higher than 5%, we go back and change things, we try to optimize the synthesis and other processes. And the cell viability, in this case, it's much better. Compared to control group, even high concentrations are very, very well. They don't damage the healthy cells. And then we go to in vitro assays. We check if the addition of folic acid, again here, increases the uptake. As you see, in both cases, concentration cases increases the uptake. And then we go back and create the groups and check their cell viability on in vitro tumor cells. And as we are trying to prove, this group should have the highest efficiency of killing cells. As you see, these are the lowest percentages. And then we go back, we don't go back, we go further. And then we go check calcium MPI dye for all these groups. As you see, again, as I want to remind, green means healthy cells, red means dead cells. So as we go this way, our treatment efficacy increases. Why? Because the amount of green decreases, amount of red increases. Because it means your treatment is killing the cancer cells. And once you put that in a chart, it looks like this. And then we go back again to the flow cytometry analysis and then check which phase, in which phase the cell division is arrested. And then we do the clonogenic assay again. And then here we have additional data, the reactive oxygen species. But I will not go into that in this chart. And then we check the survival again. And at the end, not at the end, in vitro, in vivo, this is in vivo model. Here, again, the body weight is kind of stable, and the tumor volume is analyzed over time. And as you see in control group, there is nothing done. It's just like tumor is there, no treatment, just feeding the mice. And as you see, the tumor volume like goes 3-4 times higher. And under X-ray, actually, you see a little effect. This is actually why this field is important, because right now in the clinics, there is not any approved nanoparticle radiosynthesizer for radiotherapy right now, but there is one application, I think approved or under review by FDA. It's based on hafnium oxide material, and in the next 10-15 years, we might see more nanoparticles approved by regulating agencies towards use in the clinics, because in experimentally we prove that regular X-ray is not doing a very good job compared to regular X-ray with different nanoparticles. As you see in this case, we have like the tumor volume is really low. So these are very promising and kind of exciting data. And then at the end, I want to show you, I think this is the last 2-3 slides. In general, if you want to do a complete study and if you want to understand what's happening to the whole body, you have to extract different tissues of the mice. In this case, we have mice, and check if the mice tissue is affected. Let's say you're treating the breast cancer, but is there anything happening to the spleen or heart? So what happens is that you extract, you do some histological analysis, and then you check these tissues under microscope. So the figures are kind of small here, but what I want to say is that in the breast tumor, in the first control group, you see these black dots. These black dots are cell nucleus. They are large in size, very black. But as we go to the right side here, the number of black dots are less, and their size is also smaller, which means the cell nucleus is damaged or destroyed. So this is also showing the efficacy of this approach compared to the control or the other groups. At the same time, if we check the heart, spleen, kidney, liver, we don't see any, we don't see noticeable change for these different groups. Control, even these groups between control and the treatment, when you check the nucleus and the other cells or the cell membrane physiology, whatever, they don't change that much. So this is this is something we have to do to make sure our approach is not damaging something else while it's fixing something else. This is why most of the clinical trials are failing these days, not this in the last 10, 15 years, because you generate a drug or nanoparticle or whatever treatment you have. It fixes something, but it creates another problem, maybe less or less important problem, but it creates another problem. So your approach should ideally does not have any side effects. And this is the last slide before the last slide. This is another topic that we are working on. So we are thinking, what if we put these nanoparticles, metal nanoparticles, inside a scaffold while we 3D print them? Let's say you have a scaffold and there are nanoparticles in them, and then you generate a tumor model in mice, and you cut here surgically, and you place this scaffold under here, and you sieve it back again, and you check what is happening to the tumor volume. So this is what we are working on. And actually, one criticism we got is this will never go to humans. And because once you do the surgery, why would you not take the tumor? Why would you just put something and leave? Yes, but we are claiming if this approach, and of course, applying X-ray here. So if this approach in mice reduces the tumor volume in a high fashion, in a high ratio, ideally after surgeries, after the surgeon removes a part of the tumor, removes the tumor tissue, he or she can ideally put a scaffold like this and give the radiotherapy with this scaffold. And further idea, you can also put chemotherapy drugs inside this scaffold. Chemotherapy drugs, nanoparticles for X-ray inside the scaffold. I think this is like the next project idea. So I think future can witness these studies more often. 3D printing, nanoparticles, these medical treatments are like hot topics. And then lately, I want to acknowledge the lab members. Dr. Hamed Nosrati is the postdoc I have funded through the 2232 program, and Merve, Celil, Merzi are the PhD students. And the bottom part of the slide is the master students we have. Actually, we have a medical doctor doing masters with us. He's a kulak burun boğaz expert. And this is where I want to finish. And this is the cat of, he's an undergraduate student mastering in food sciences. And where I want to end the slide. This is the contact information. I want to add this in case people find this interesting and want to reach out or follow. This is the information I want to provide them. And this is where my presentation ends. Thanks to all for listening to me.
Thank you, Professor. It was very interesting. I was just taking a course this semester about radioactivity, and it was like seeing these things that I studied coming to life. So it was very interesting. And I have two questions for you. First one, how do you target cancer cells with nanoparticles if the healthy cells also have folate receptors in their surface?
Yeah, this is a good idea. Whoever asking this question knows what's going on, because folate receptors are also found in healthy cells, but to a lesser extent. Which means tumor cells, if they have like 100 receptors, I'm just making up the numbers, 100 receptors, maybe healthy cell has 10. So which means your particles are still going to where you want more than where you don't want. But this is another field of research, finding the receptors for each cancer cell. Type of cancer cell. So each cancer type has other receptors as well. It's not just folate. We used folic acid, folate, because it is well known and it's the first approach we thought of. And they say there is Herceptin 2 receptor for breast cancer, some other receptors. You can ideally put molecules outside your particle. You don't have to do just one. You can do a couple of different types of modifications so that your particles go where you want to a more even higher extent. So this is like, I'm not presenting as if like, oh, we solved the problem and bye. No, this is just the starting approach. This is just the first steps of a baby. I think people will take over. Maybe we will work on this throughout our lifetime. But this is an exciting field and open to explore a lot. And I want to say, maybe we have students from biomedical engineering departments. I think the smart thing to do is focusing and chasing the future topics and hot topics and futuristic ideas, not studying the 50, 100 years old as well established topics where you cannot generate something new easily. Maybe I diverted from the topic a bit, but this is my personal idea because I think most of the listeners are students. I think this is important. And there's no question, but let me add my idea. Getting a fundamental education about physics and chemistry is very important. When I go back, if I had the chance to go back to 20 years old, I would study physics and chemistry in a fundamental way, because most of these things, there's synthesis, there's X-ray, there's physics and chemistry inside between each other. You know, you have to, if you want to really do some impact, you have to have a solid fundamental background. Okay, please. Second question.
Yes, I have a question too.
Yeah.
May I ask a question too? By the way, that was very informative. That was very interesting topic. And I see some of my good friends in your project, two Ömers. Yes. One of them is my best friend. The other one, I work in a 1001 project from Erciyes University. So it was very good to see them too. My question is, I see after the dead and viability assay, some of the cells, a quite significant percentage of cells remain alive. So are you like, do you wonder, or do you ever worry about provoking cancer stem cells if they are the ones that are left over after the nanoparticle application?
In vitro, right? In vitro experiments?
Yes.
I mean, it can be done. If we can dye them, we can, maybe you can help us with that. If there is a way to show them in a different color or somehow depict the pictures, the stem cells. Yeah, I think that's very meaningful question and idea.
But do you worry about, you know, provoking or triggering them, creating a worse scenario, you know, because the cancer stem cells after the radiotherapy or chemotherapy, they become more aggressive. So do you
maybe in the long term, they may tolerate the radiotherapy, they may develop a radio resistance. That's why after surgical removal of tumors, most of the time or a large number of time, chemotherapy and the radiotherapy are applied together. Most of the time chemotherapy alone, but sometimes they are applied together. But you know, the cancers, maybe in the long term that might be a problem, and there has to be some other mechanisms to reduce the resistance of cancer cells against radiotherapy should be sought. But the data we have clearly says it has a distinctive effect, positive effect. The addition of X-ray to these heterostructured nanoparticles clearly doing something beneficial, but it can be further improved by maybe going into details like stem cells, cancer stem cells.
Great work. Thank you. Farida, do you have more questions?
Yeah, I have a small scale question. It's about the, you mentioned something about in vitro, about the cell phase, and some phases are more sensitive to radiation than others. Do you target a specific phase, or do you just hope that it's in the sensitive phase? And if yes, how do you make sure that all the cells are synchronized?
To be honest, this is something I don't know very well, but we try to have high percentage of arresting the cycle in G2 and M phases. So if you ask me why these phases, like, and how, I also don't know very well. Like, maybe this is, I don't know if there is an answer actually. Maybe Esra Hocam can give an answer if she knows.
I think she's asking if you have a chance to synchronize the cells at the same level, for instance, leaving them like hungry, for instance, leaving them without a serum, so they be on the same stage before the application.
Maybe in vitro you can try things, but in vivo,
right? Yeah,
it's
of course.
not. It doesn't look very feasible, right? But in vitro, maybe you can direct them to a certain phase. But these are not any, the data I show, the 29%, 30%, these are like nothing done, just particles put there, X-ray applied, and that's what percentage. If it goes to 50%, it's better, but how to take it to 50%, not giving enough medium, culture medium, but then it will decrease their size. Maybe they will start dying without even X-ray. So I'm not sure about it.
That's
But
right.
these
That's
are
right.
really good questions.
Like,
they give me homeworks.
usually what we do is we might, just an overnight experiment. We just leave them without, FPS medium, but no FPS. So they can be hungry for the next day, but all of them at the same stage. You might want to try that maybe.
This can happen in vitro, but what about in vivo?
you can starve them one day.
Starve?
those little mice.
Maybe after radiotherapy or before one day, don't eat anything. Maybe it works better. I don't know. I'll check these. These are important questions.
Okay.
Thank you, Professor. It was a very interesting talk.
I thank you for inviting me.
Ne kadar geçmişi Bruni Üniversitesi'nin kaç yıl oldu Hello, everyone. This session that we will start with Professor Dr. Mr. Shaheen will be the last speech of the day and the Congress. I would like to thank you, the participants who have so far aspired to our Congress with great enthusiasm. Before starting his speeches, I would like to give you some information about Professor Mr. Shaheen. Mr. Shaheen received the bachelor's degree in electrical engineering from Istanbul Technical University in 1986 and master and PhD degrees in biomedical engineering from Case Western Reserve University in 1993 and 1998. He has been on the Faculty of Biomedical Engineering at New Jersey Institute of Technology since 2005. His research interests include floating microstimulators and neural recording techniques from the spinal cord and the cerebellum using flexible multi-electrode arrays. At the end of the professor's speech, you can send us the questions that you want to ask our esteemed speaker through the chat section. Now I leave it to Professor Mr. Shaheen to deliver his speech on non-invasive modulation of the cerebellum using ultrasound and transcranial electric stimulation for treatments of neurological disorder. Please welcome, Professor.
Hello, everyone. I hope everybody could see my slides right now.
Yeah, we can see.
All right. First of all, thank you all for inviting me to this nice platform and allowing me to connect my colleagues in Turkey, my home country, and I commend all the organizers for this opportunity. So I understand the target audience is undergrad students, and therefore I will try to speak slowly and make it hopefully as clear as possible. My area of research, as Ahmed mentioned, is neuromodulation these days. I started out my career as a neural stimulator person in the peripheral nervous system, and then I moved up to spinal cord, and now my most current interest is in the cerebral. Let me introduce NGIT a little bit. We are in New Jersey, across the Hudson River from New York. This is where the administrative building is. Our new athletic center. Our building is here at the corner of the campus. NGIT has been growing and is increasing in ranking nationally, etc. We have the department is about 20 plus years old. As far as the undergrad program goes, before it was a graduate program. We have about 400 plus students in the undergrad and 50 PhD students and as many master students. I am also the director of the PhD program. So with this introduction, I like to continue. Before I get into my topic of today, let me introduce you, let me cover a few slides about the projects that I have had in my lab for the past 15, 20 years, etc. Floating light activated microelectrical stimulators, flames, as we call it, was one of the projects that we pursued for many years. These are very small, sub-millimeter size devices that could be activated using infrared light. And our intention was to implant these into the spinal cord and in the most mobile parts of the nervous system. And by shining light, we could activate these devices and they generate electric current in order to stimulate neural tissue. You see the students here who were involved in these projects in some of these slides. So in this project, we developed these chips, little chips implanted into rats, spinal cord, and by shining light through the skin, through the tissue, you could see that the limbs are moving. This is the hand closed before stimulation, and this is the hand during stimulation. I have a little video here actually to show how the movement occurs during this stimulation. So all in all, we've shown that this was one of the first projects showing the feasibility of wireless, electrically, optically activated electrical stimulators, microstimulators, could be implanted into the nervous tissue and generate a motor response. So the idea is, of course, in neuroprosthetics, you have to implant things into the nervous tissue, brain, spinal cord, what have you, in order to inject this current. And it's an invasive technique. In most cases, clinical translation is quite difficult because of this invasiveness. Here, the idea is if you could do the same using wireless optical stimulation, the neural tissue will experience much less trauma. Especially these devices are floating, meaning that as the neural tissue moves around, the device also moves. So that minimizes the neural tissue damage, as it might be induced by the connecting wires with the traditional devices. So we showed the feasibility of this device, and we got NIH funding and several publications, etc. The next project, a major project that we had in the lab, was the spinal cord computer interface, which is for the spinal cord injury subjects. Obviously, they do have action potentials, neural signals, controlling the periphery, coming down the spinal cord. But after the injury, those signals are not able to come down and reach the muscle. So depending on the level of injury, the person is paralyzed from that level down. The original idea was, this was an original idea proposed by myself years ago as a postdoc, actually, at Case Western Reserve in late 1990s, to access the descending signals at the level above the injury and record these signals into the computer and acquire the evolutional signals from the spinal cord. This is a similar idea that you might have seen this movie, Spider-Man 2, where Dr. Otto has these spikes going into the spinal cord, and in a few seconds, he starts controlling the robotic arms. Of course, nothing is that simple. The spinal cord would be terribly damaged if you do what he did, but there are ways to interface with the spinal cord. And I have to claim that we proposed this idea before the movie came out. So NIH also started this program here, Bridge the Spinal Cord, a few years ago, exactly proposing the same idea, receiving proposals from the community. Our next interest in the lab for the past 10 plus years has been the cerebellum. Cerebellum is an amazing structure. This small structure here at the back of the brain actually contains more cells than the rest of the brain. The cells are just more impact and closely packed in a smaller space, and it has a very stereotypical, highly organized structure. These are excuse me. Excuse me. These are microsomes, as they call it. They're like almost parallel processing units. There are several cells involved in this circuitry. They're all, this similar structure is repeated across the cerebral cortex. And cerebellum is involved mostly in the sensory motor control. There's the the idea proposed for the role of the cerebellum is that there's an internal model. There's a forward model and there's an inverse model that predict the results of our actions. So if you think of moving your arm, and this intention goes to cerebellum before even the movement begins, and the cerebellum can predict the results of that action either in the form of motor signals or sensory signals. And then the cerebellum fills in the gap. It compares the intended movement with the actual movement and does correction on the fly. This is how we play tennis, for instance. But any kind of ballistic movements that we do, playing tennis or shooting arrow, anything that is ballistic, which means during the action, we do not have the brain, the central nervous system doesn't have time to make corrections. So it's the cerebellum's job to receive that information and make sure the resulting movement is as perfect as possible. So there is an internal model inside for every action that we do repeatedly. Of course, there's a training period, there's the learning period, I should say, during which time these internal models are built, and then cerebellum starts using these models to help out or make our movements, fine movements especially, more smoother. So I was interested in the cerebellum because it's involved not only in sensory motor coordination, but also as shown in recent data in all kinds of other functions that the brain is involved, all the way from emotions to cognitive functions, etc. Obviously, then involved also some neurological disorders such as Parkinsonian disease, tremor. There's the epilepsy, there's the cerebral ataxia, depression. In fact, these days, people are looking at the cerebellum from all kinds of different neurological disorders. So as a person who is trained to modulate neural cells, I was interested in looking at the cerebellum, how we can use this structure and our knowledge in neuromodulation to produce some treatment for some neurological problems. So first, we started out by looking at the cerebellum with some recordings we made in rats to see if we can acquire signals, if we can see their correlation with the motor function. So we used some of these high-tech surface electrodes that can be put on the cerebral surface. These are really small, 1 or 2 millimeter wide devices with micro contacts, and recorded the signals while the animals are in different states. And we saw a striking difference between those recordings we made from the cerebellum versus the ones we made from the motor cortex. These plots here showing that during anesthesia, with the cerebellar activity, this black one here, brown one, during the anesthesia, this activity level really goes down compared to the wakefulness. Whereas the recordings we made from the motor cortex is already very low at those high frequencies. We couldn't record anything. Cerebrum is the motor cortex, actually. We couldn't record anything beyond 150 hertz from the cerebrum, while the cerebral activities were going all the way up to 800 hertz or so. That is because if you go back to this structure, there are Perkins cells, as they are called, very close to the surface. These are the output layer of the cortex, actually, in the cerebellum, which is very close, like a couple of 100 micron below the pia surface. And these are very active cells. and they form, as I said, the output of the cerebellar. After all this computation, the Purkinje cells carry the information, process the information, and send it down to deep cerebral nuclei. So in the recordings that we receive, we collected from the surface, those are heavily biased or contain activity from the Purkinje cells. That's why we could record these high frequencies. So in general, this project here conducted by these students indicated that yes, cerebral activity can be recorded, and it's highly dependent on this state of the animal, whether they're anesthetized, resting, face cleaning, or highly active. The cerebral activity was changing tremendously. Another student of mine who's actually still working on this project, hopefully graduating this summer, has looked at the correlation of the cerebral signals with the, movement of the forearm. The animals are trained to reach through a window and grab a lever, a force bar, pull the force bar, and then of course they get a sugar pellet as a reward. While we are recording the neural signals from the cerebellum and also EMG signals from the forearm. And these are showing the small carbon electrodes that we use. It's SEM picture here on the right side. Is showing a 7 micron thick carbon electrode, which is coated with some insulation material, of course. And you can imagine this is like 10 times smaller than human hair. And they're connected to some microconductors using some silver epoxy in order to be able to make this bundle. And we insert these bundles into the cerebral cortex to record this neural activity. This is a new technique that some labs are pursuing, the carbon electrodes, I mean. So we looked at the neural signals and the EMG signals, and you could see here high correlations for different muscles, dorsal side of the arm, ventral, another muscle from the ventral side of the arm, biceps and triceps. So also significant correlation with the cerebellum, which means that the cerebellum is involved in coordination of these movements. The original idea, again, in this project proposed by ourselves, was to use cerebellum as an alternative to brain, current traditional method of brain interfacing. Current brain interfaces are using signals recorded from the motor cortex, or mostly sensory motor cortices, in order to extract the evolutional signal of the person. So you make an intention to move your arm. However, the person is paralyzed, so the limbs cannot move. However, those signals extracted from the brain can be used to control, for instance, a robotic arm. Or in some cases, you can use these signals to trigger electrical signals that will be stimulating the paralyzed muscles of the person so that they can regain motor function. In this case, the idea is, instead of using the motor cortex as the source of evolutional signals, to use the cerebellum because of its involvement in these motor behavior. The cerebellum is known as the processing of mostly sensory signals, but it is anatomical data showing that cerebellum actually has lots of inputs, actually millions of cells, neural pathways coming down from the motor cortex. So there is the evolutional component of these signals. And in fact, the student on this project, Esma, she moved when you move the neural signals with respect to the EMG, if you can delay them in the positive and negative direction, you can come up with some causal relationship. If the correlation is higher when the neural signals come before the EMG, you can tell that the the AMG signal is the result of the neural signal. And that's what we've done, and it's shown that this is a causal relationship caused by neural signals caused by the, EMG signals caused by the neural signals. So there is the motor component, more so than the sensory component in these signals. This is an ongoing project, and hopefully a publication is coming out soon. So now I'm gonna switch to, after these couple of recording projects from the cerebral cortex, I'm gonna switch to neuromodulation idea. Transcranial electrical stimulation has been around probably for a couple of decades, but recently it came to the attention of researchers because it shows that you could actually modulate neuronal function non-invasively in quite a different spectrum of disorders. There's different forms of transcranial electrical stimulation. There's the DC stimulation. I'm interested in AC. There's also noise type of or random electrical stimulation as well. Here, in a nutshell, you have large electrodes across the head, applying a small milliamp level currents to the entire head, actually. And so in some forms of this stimulation, the electrodes could be smaller and more local, but the idea is transcranially, even through the skin, you can inject this current and modulate the neural activity. And of course, because the AC current changes directions, depending on the frequency, you may be able to target different neuronal functions and different neural circuits. I like to emphasize a fundamental difference between what we call suprathreshold and subthreshold modulation. Up to maybe 10 years, 15 years ago, in this field, people in the neuromodulation area, when we say neuromodulation, neurostimulation, we always thought of suprathreshold, where you apply an electrical pulse, mostly extracellularly, to the neurons, and then an action potential is generated in the neuron. The spiking activity is generated because of this immediately after the pulse or even during the pulse. This is a suprathreshold. There's one-to-one correspondence with the stimulus at the action potential. Now, the subthreshold stimulation is a different ballgame, where you apply this electric field to a whole bunch of cells. And depending on the orientation of the cells and how much electric field they experience, the cells may be, of course, hyperpolarized or depolarized, or different compartments even of the same cell may experience depolarization or hyperpolarization. Here, the idea is we're not actually inducing an action potential in a one-to-one type of relation. We're only moving the transmembrane potential in one direction or the other. If it is, there's this spontaneous activity, therefore, in the cell. The cell is firing on its own. Most cells in the central nervous system, they do fire action potentials spontaneously on their own. So there's this ongoing activity. And by applying this hyperpolarization or depolarization, you are increasing or decreasing that spontaneous activity. In my opinion, in the future, this type of stimulation or modulation paradigm will find much more applications than the traditional suprathreshold, because in many disorders of the central nervous system, there is this overall network activity is just aberrant and abnormal in slightly shifted in one direction. All we have to do is just push it back into the normal state, rather than just inducing and controlling that circuit using suprathreshold stimulation. We can just push it in the normal direction, and while keeping the spontaneous activity, because spontaneous activity in the CNS carries crucial information, especially in the cerebellum. In the cerebellum, even the timing of the spikes are important, not just the frequency of spiking, but even the timing of the spikes are very important. So if you overwrite that information by applying suprathreshold stimulation, you're losing that information as well. Instead, you could just correct whatever is wrong. So this is, in general, what the transcranial stimulation does, because the electric currents are very small and they provide a continuous depolarization or hyperpolarization in the cell. So first of all, we wanted to see what kind of electric fields can we produce inside the cerebellum or the brain in general. We applied an electric pulse. In the rat, we positioned the electrode at different levels on the skin, on the skull, or on the dura mater to see how far the electric field can penetrate. And interesting enough, the electric field, the vertical electric field, not the voltage, but the electric field, which is the voltage per centimeter or millimeter, declined very quickly. Within a millimeter or two, the amplitudes were decimated, which means the data is very noisy, as you see, because there's the heterogeneity in the neural structure. These are direct measurements inside the neural tissue. So there's the variance due to different anatomical features of the neural tissue. But in general, the E field was declining very quickly, which means in all these applications, whether human or animal, the E field will be maximal near the surface. So before you can do anything inside the brain, like a few centimeters down from the skull, you will be modulating and affecting the cells that are very near the surface. And in fact, you may be driving them too fast or too strongly to the suprathreshold level. This is something that everybody in this field should be aware of, and they are. But this data, I think was the first one to show that with real animal data, experimental data, that the decline is very quick, and confirm the computer simulations that some other labs have been publishing. You can also derive these E fields using models like this, analytical equations that have been around for decades. And in general, they agree with our experimental data. However, there's a significant difference because no analytical equation can really perfectly simulate experimental data. So then once we have this better picture of how we can, what the electrical field looks like inside the brain, we went ahead and used these custom-made electrodes to apply electrical fields to the cortex of the cerebellum in different directions. So this is a novel design. We applied the electric field either in the medial-lateral direction or rostrocaudal direction, or in the perpendicular to the plane of this electrode, which is the dorsoventral direction. So in 3 orthogonal directions, we were able to produce electric fields in the cortex. This is showing here a recording from the cerebral cortex. Perkins cells, in fact, these large, they are the largest cells in the cortex, the black large cells. This is showing simple spikes of the Purkinje cells. They are known to produce also what's called complex spikes. Complex spikes coming like every second or so. They're supposed to be the correctional error informing type of spikes, but simple spikes run anywhere from 50 hertz to 100, above 100 hertz in the Purkinje cells. So we applied this AC stimulation to the head, to the cerebral cortex using this type of electrode. And as you see in this spiking activity, there's clear correlation with the AC signal. The amplitude of the AC goes up, the spiking frequency increases, and vice versa. So we were able to modulate directly the Purkinje cell activity at very low electric fields, a few millivolts per millimeter. Clinically speaking, now those who are doing the clinical work, they like to keep the electrical field below 1 millivolt per millimeter. We applied a little bit more to see this clear modulation. But the same thing happens at the lower amplitudes as well. So here in this slide, we're showing modulation at different amplitudes. As the amplitude is increasing, clearly the modulation index, as we call it, which is the standard deviation over the mean of interspike intervals, is increasing as well. These are complicated slides, but I will tell you the take-home message. In 3 different directions, we applied the field, right? Mediolateral, rostrocaudal, and dorsoventral in 3 orthogonal directions. And we found that the highest modulation was possible with the rostrocaudal direction, which is the direction in which these Purkinje cells are oriented. They are in a rostrocaudal plane like this. They are in parallel planes. These dendrites of these cells actually contain within a very narrow rostrocaudal plane. So if we apply the electric field in the same direction, it seems that we were able to modulate the activity, but in the other 2 directions, it wasn't as effective. So that's a very important finding for the clinical application, where these electrodes could be actually put bipolarly across the cerebellum in a certain direction in order to maximize the effect. We also tried different frequencies. Here you see going all the way from 2 hertz to 100 hertz. The interesting thing is, as we increased the frequency, there was almost one-to-one correspondence. Every AC cycle was containing or causing 1 spike. So that's called entrainment. We're not stimulating the cell at the suprathreshold level. We're only changing the transmembrane potential, but the cell spontaneously firing at those specific times that are controlled by our AC stimulation. So, of course, such a strong entrainment may not be preferred in the clinical applications because, again, you are overwriting the information that is already there. But at the lower frequencies, you can actually synchronize these activities. And I can speak to that maybe in the later slides. Synchronization among the population of Purkinje cells is a key factor in the cerebellum in the processing of signals. The synchronization amongst the Purkinje cells means that the next level of cells, which are the deep cerebellar nuclei cells, can take that information. A disordered spiking of the Purkinje cells actually do not get forwarded to the next stage. These are histograms, but essentially it's the same data here showing in a different form. And we also made an observation that If you increase the frequency too high, the cells begin skipping, meaning that they're not going to fire at every single cycle, but they will skip the cycle. They will skip every other cycle or every 3 cycles of the AC signal, etc. We also applied a DC, which is also very common these days to test in the clinic. I mean, in the research phase still, but you're gonna actually buy one of these on Amazon or eBay, etc. Interestingly, you know, because the current levels are so small, it's not supposed to produce any harm, and people can test these at home, which could be dangerous if you of course, you're not allowed to increase the current, so it should be safe, but they're available basically even without the consent of a doctor, which is a different story. Here we tried DC modulation. We applied a DC current for about 20 seconds or so, and we saw a clear difference, increase and decrease in the spiking activity. This is the vertical axis here, the spiking frequency of a single Perkins cell. As soon as we apply the DC, the spiking goes up, the spiking frequency goes high immediately almost, and then there's an adaptation period. It slowly comes back down to the baseline by itself. This abrupt change actually in the spiking frequency suggests that, you know, maybe in the clinic, in a human subject, we should not apply the current. The onset of the current should be smoothed somehow with a transient, slow change, because this abrupt change in neural activity can cause a startling effect. The subject may experience some different, things that are not very, very pleasant. It will be startling to the subject. So in order to avoid that initial response, maybe the current should be applied slowly, which is today actually what most people in this area are doing. But this is confirming with the animal data that the change is immediate, very quick, and that should be avoided. Now, maybe it's a little bit too late, but let me go back to the cerebellar anatomy once more and emphasize one important thing here. This circuitry, as I said, repeated across the cerebral cortex, but a given module consists of only a few different type of cells, and the biggest player here is the Purkinje cell in the middle, PC. And that cell receives all that information through parallel fibers and other cells, basket cells, satellite cells, granule cells, etc., collecting that information and produces one output. That output goes to the deep cerebellar nuclei. And from there on, it goes to the rest of the brain. The cerebellum is sending these projections to all across the brain, not just the thalamus. Through the thalamus, it goes to many other places, including the sensory motor cortices, of course, but even the amygdala, for instance, which is involved in the emotions, etc. So here, our main purpose is to modulate the Purkinje cells in order to be able to modulate the cerebellar nucleal cells, in order to see some behavioral effect. So here, I'm showing a slide where we applied this AC stimulation to the cortex in the rat. This is a picture of the back of the head, where the cerebellum is, and this wire, which is silver chloride wire, is simply touching on the cortex with a circular area. And when we inject the AC current, we see a modulation. But let me first emphasize that we are recording from the cerebral nuclei, not from the Perkins cells. Perkins cells would be closer to the surface here. There's just a few millimeters, 4 or 5 millimeters difference, but that's a long distance in CNS. And here you are seeing these spikes captured with our electrodes. These are glass microelectrodes, which is one of the oldest techniques in neurosciences. You take a glass tube and pull it from both sides while heating in the middle, and when it breaks, it makes a very fine tip all the way down to a micron size. And you fill the glass electrode with some conductive solution such as KCl or even saline in order to provide this connectivity, electrical connectivity, to record from single cells. And that's the technique we use to record from this Perkins cells. You see these hundreds of thousands of spikes detected and shown on the same scale. And when we plot a histogram, here histogram means the probability of firing of a cell with respect to the cycle of this AC. So AC cycle here in this case about 10 milliseconds, which is 100 hertz. And as you see, the firing frequency, firing probability is much higher at certain phases of the signal, which means we are actually modulating it. Without this modulation, this will be a flat histogram. So we can, of course, quantify this by defining a modulation index, like the covariance of this change between the covariance of the signal between the histogram and the AC, and divided by the mean value gives us a number that is somewhere between 0 and 1. And here the modulation index is about 0.5, 0.4, etc. You could also categorize these action potentials to make sure that you're recording from one cell. There could be multiple cells in this recording. And here there's a 2D plot of the cells, amplitude versus duration, and they're clustered based on the duration and the amplitude. And you can pick a certain cluster which will correspond to a unique cell. So here's the summarizing slide of this experiment, where the frequency and amplitude of modulation has been varied. On the horizontal axis, we have the frequency going all the way from 1 hertz to 400 hertz. And you cannot appreciate it here, but the amplitude is also changing from the bottom plot to the top plot. The top plot has the highest amplitude. But the take-home message is that there's a range of frequencies where the entrainment is maximum or most effective, such as like between 100 hertz and 300 hertz, you are having the highest modulation index. But below, especially 50 hertz, modulation is very different. Entrainment, I should say. So here we're recording from the cerebral nuclei, not from the Purkinje cells. However, the AC is applied to the Purkinje cells on the surface. We're modulating these Purkinje cells, but because of the synchrony amongst the Purkinje cells, we are able to affect the spiking activity in the nucleus. And from the nucleus, as I said, the projecting activity goes to the rest of the brain. So here's a direct evidence from animal data that not all the frequencies are good. Yes, the higher the amplitude, the higher the modulation index. However, certain frequencies are better, especially frequencies higher than 50 hertz and 100 hertz. They are much more strongly able to modulate. Why is this important? Because we want to minimize the electric field. If you increase the electric field, to the above the limit, we may be actually causing suprathreshold stimulation, or at least the cells that are very near the surface will be affected much more than the others, and there could be some undesired results. Here, with a minimum current, these are like less than 100 microamps. Of course, because in order to avoid and eliminate all the other complicating factors, here we open the skull and apply the current directly on the cerebral surface. But in the clinical setting, the current will be applied transcranially, actually transcutaneously across the skin. But yet, again, minimization of the current is the ultimate goal here. And it seems that in order to minimize the current, the higher frequencies are preferred. Chi-Kang has been the student on this project and has been very productive. Now, as my title of the talk says, we have tried a similar approach using ultrasound, which may also be quite interesting for some of the audience here, because ultrasound is a new paradigm being tested. And it's very attractive because not only it's non-invasive, but also it can be focused, unlike the electrical signals. Electrical signals, when you apply through the electrode, it spreads all over. It's highest in amplitude near the surface, but there's no way to focus it at a depth. Ultrasound has this advantage because there's the propagation involved, and there's the interference pattern that you can produce inside. So ultrasound, these acoustic waves are producing pressure field inside the neural tissue. And that pressure field has been shown to modulate the neural activity. These are just commercial devices that you can purchase. Those are called immersion type ultrasonic probes. The frequencies we're talking about is in the range of hundreds of kilohertz up to a few megahertz. And they can be focused, as shown here, at a depth of, let's say, several millimeters up to several centimeters. Our colleague at North Carolina State University, Dr. Oral Khan, has helped us to produce these pressure maps using hydrophones. Hydrophones is a sensor that can measure the amplitude of these pressures as a function of space. Here's the X and Y coordinates. So we're looking at a cross-sectional area of the field, and this is a transverse cut in the orthogonal direction to the axial direction of the field. It's a circular, it's like a sausage type of field, as shown here. Up here, it's deeper than the horizontal direction, but it's focused after all. And it penetrates several millimeters into the tissue. The yellow areas are the highest pressure areas. And first of all, using a cone, like a 3D printed cone here, a plastic cone, we could focus it further because the focusing depends on the frequency and the propagation speed of acoustic waves in the tissue. And you cannot go down below a certain size depending on the frequency. This was a 500 kilohertz transducer. So the smallest focal point we could get was like in the order of a few millimeters, but with the help of a cone, we could make it smaller, even though the field is no longer very smooth. There's a high pressure field in the middle. So again, just like we did with the transcranial electrical stimulation, we applied this ultrasonic signal to the surface. while recording from the Perkins cells near the surface, right underneath the surface. And Perkins cells has this characteristic spiking activity. These are simple spikes again. These are the complex spikes, as I called before. So here's a recording. Ahmed was the student on this project. He's now a postdoc at Columbia. He recorded these signals when the ultrasonic pulses were applied, 0.5 or 1 millisecond long. It comes in as a train. We applied train, and there's a spike action potential right after each ultrasound stimulation at 50 hertz or 100 hertz. And we could again do it's called peri-event histogram, which means you're triggering the plot by the focus ultrasound stimulation, therefore showing the correlation. And you get a nice histogram like this. It's like the probability distribution function, where it shows a high probability following the stimulus, ultrasonic stimulation. Without the stimulation, it's almost flat. With the stimulation, you get this high peak right at the end, or with this little delay after the ultrasonic pulse. So we did this for different durations of the ultrasonic pulse and showed that short or long pulses, regardless, the stimulation occurs almost about a millisecond after the onset of the ultrasound. I'm sorry about my voice. As I said, I have some allergy symptoms this morning. Excuse me. So here, again, we wanted to also measure the CN, this cerebral nuclei activity, to see if we are doing anything to it by modulating the Perkins cells. The CN here is a few millimeters deep from the surface, and we could penetrate with glass electrodes while applying the ultrasound to the surface. And clearly, the activity of the CN was also modulated nicely. I forgot to mention that the projection from the Perkins cells to the CN is inhibitory. So higher activity actually in the Perkins cells means lower activity. If the frequency of firing in the Perkins cells are too high, then it will completely block the cellular activity in the nucleus. And that's what we are seeing here. During the ultrasound stimulus, the activity is completely inhibited. I'm gonna fast forward here a little bit because I think I'm losing my voice. And in a nutshell, we have done the same thing with the ultrasound that we were able to do with the transcranial AC. In fact, ultrasound can also produce some local field potentials. Local field potentials are these signals that are below 300 hertz or so, and they're associated with the population activity, mostly ascribed to synaptic activities. So here, if you look at the frequency spectrum with and without the ultrasonic stimulation, there's a difference in power spectrum, indicating that during the stimulus, which is indicated by this pink bar, there's also low frequency changes, not just the spiking activity, but also low frequency changes, showing that we are doing something to the transsynaptic activity. Let me mention also the synchrony. Why is synchrony so important? This group published this paper almost 10 years ago, which is very interesting, showing a very interesting phenomenon, that when the Perkins cells are just firing randomly without any synchronization, the deep cerebral nuclei is not modulated. When they are firing at a high frequency in a certain time period, they can block the nuclear activity completely. But when they are synchronized, more or less, the synchronization level doesn't have to be perfect, but even if when they are partially synchronized, then they tend to entrain the activity of the cerebral nuclei. In other words, the nuclear cells stop following the firing frequency of the Purkinje cell. These first three rows are Purkinje, and the last row is the nuclear cell. So in the other two up here, there is no correlation or similarity between the firing patterns of the Purkinje and the nuclear cells. But in the last one, there is entrainment. The frequency is the same, and it's following the same pattern. So that's our intention by providing or enhancing the synchrony among a population of Purkinje cells. We like to be able to modulate the nuclear activity. And the reason is, in both paradigms, I mean, with the transcranial stimulation, of course, the surface is more affected than the deeper cells. But with the ultrasound, we should be able to do both. We should be able to either activate the Purkinje cells and thus indirectly train the nuclear cells, or directly modulate the nuclear cells. So, in conclusion, yes, we were able to show this modulatory effect, both using transcranial AC and ultrasound. Transcranial AC, in fact, indicated there's a range of frequencies that is favorable. Higher frequencies are better. In fact, there's now some clinical data showing that high frequencies are more effective in human subjects. And also, we've had this animal experiments showing that the electric field actually doesn't penetrate to a great extent. But what's the big picture? Again, I just would like to remind you, the big picture is spiking entrainment of the spike activity in the cerebral nuclear cells can actually affect the behavior, whatever that behavior is, whether it's a mental activity or some aberrant activities such as epilepsy, Parkinson's, tremor, cerebral ataxia, or some, other forms of neurological disorders, mental disorders, depression, dementia. And there's a paper came out, I saw, that they are trying to wake up the comatose patients using ultrasound, focused ultrasonic stimulation. Because those are non-invasive techniques, its clinical translation is much more favorable compared to the implantable electrodes, and that's our ultimate goal here. So let me also introduce some lab members. These are some of my previous students who are at different places now, anywhere from industry to academia. And these are some of my current students in the lab who helped me do this research. And we have support from mostly NIH. I work with mostly NIH. Thank you.
Teşekkürler hocam. Thank you hocam. It was a very nice, informative talk. I just have one question before Ahmed. I think Ahmed has a question. I was wondering if you could use higher frequencies for brain tumor cells to destroy them rather than. I'm sorry?
So in general, in ablation of tumor cells, or the idea is the ultimate goal is to stop them from reproducing, right? The focal ultrasound will have an effect of course heating effect. If you crank up the power, it will tend to increase the heat in the local area, and that's a technique that is being used to ablate tissue. So if that's what you are leading to, yes, it can be used to kill tumor cells. Here, of course, the objective is completely different. We're not trying to kill anybody. We just want to modulate their activity.
I know the reason I'm asking because I'm a cancer biologist, so I was wondering if I can use the same technology, but in a reverse motion to you know, to advantage my work. So
I mean, increasing the activity in a local area where the tumor cells are, how would that be? You know, how would that affect the growth of the tumor cells?
I was just wondering, maybe we can, you know, make a change on the membrane and cellular membrane because of this electrical change. Will
Yes, I mean that could be
might be more sensitive to the chemotherapy, maybe.
Maybe.
Okay, that's what I
In
was
fact,
doing.
there's initiatives within NIH in the US to combine these multiple therapies, multiple modalities in order to produce some unique modulation paradigms that are not possible with individual stimulation techniques.
Thank you so much.
Thank you very much for a wonderful presentation. And I believe we only have one more question left. And the question is, what kind of damage does it give if we use invasive modulation instead of non-invasive?
Invasive means you have an electrode that is inserted into the brain. There are microelectrodes and macroelectrodes. In the case of deep brain stimulation, for instance, the electrodes are macro, meaning they're like a millimeter in diameter. It's a metal rod, basically, that is pushed into the brain all the way down to the area of basal ganglia. However, of course, on the way, it damages tissue and neurons, but it produces the desired effect. And this partial damage in a few cells, you know, clinically, it does not matter. At the targeted location with this macro electrode, you're able to stop the activity of some aberrant activity and stop tremor. And it has tremendous benefits clinically. So for stimulation, that's okay, because you can always, even though you damage or kill some of the cells around the electrode, you're still able to push current and activate the other cells that are a little bit further away. But with microelectrodes that are in the order of a few hundred microns, maybe, or less, there will be encapsulation tissue, no matter what, because you're introducing a foreign material to begin with. And then if there's any micro motion of the electrode, that will even cause fibrous tissue in the long term. And yes, stimulation would still be feasible because you can always increase the current. However, now recording becomes almost impossible because this fibrous tissue is going to push all the live cells away from the electrode. So with microelectrodes and macroelectrodes, the tissue response, minimizing the tissue response, is one of the important aspects. That's why there's a whole bunch of research going on in order to test different materials, different electrode geometries, different implantation techniques to minimize the trauma. But going back to your question, it is still, you know, for brain computer interfacing, for instance, what Elon Musk is doing right now, right? He's intending to put not just a few hundred, but thousands of electrodes into the brain in order to go to the next stage of this brain-computer interface. This is, by the way, not his invention. There's hundreds of people who paved the way to this state of technology, and he's just recruiting these highly skilled individuals in the field, and hopefully trying to take the project into the next level by increasing the number of electrodes by a factor of 10, maybe, etc. But when you put so many thousands of electrodes, obviously the neural damage becomes even more important. That individual electrodes, if you can find the kind of material, type of electrode geometry that induces the minimal damage, then that will really open up the possibility of increasing the number of electrodes even more.
Okay, thank you so much for answering our questions, and thank you for participating in our Congress and sharing your valuable information with us and giving us new perspectives. We appreciate it, sir.
My pleasure.
Thank you so much, Hocam. We hope to see you again.
Same here.
All right, everyone. Folks, thank you for listening our nice presentations and joining us during the day on a very beautiful Saturday in Istanbul, which we haven't seen for a long time. So thank you so much, and we hope to see you with another event. Ahmet, thank you so much for preparing everything, and you
No
guys,
problem.
Biomedical Engineering Department Club, thank you so much.